Thank you to the organizers for inviting me to this talk. This is the title that I had for this talk, I guess probably last February, so at this point that's about 11 months ago. Much of this talk is going to be based upon this figure, or at least what I've derived since then, since publishing this figure early in 2016. A more appropriate title for this talk would be Phage Delay Towards Enhancing Bacterial Escape from Biofilms, a more comprehensive way of viewing bacterial resistance to phages. And this is something that, with luck, is going to become better appreciated in the course of me giving the talk. So what I'm going to be talking about are imperfect mechanisms of resistance to phages as affected by bacteria. So by imperfect, I generally mean not fully functional. So I'm not talking about restriction modification systems or CRISPR-Cas systems. I'm also not going to be talking about mutations that prevent phages from actually adsorbing to the surfaces of bacteria. Instead, I'm talking about essentially everything else. And this is going to be mostly everything that people think about well, not including everything that people think about when they think about bacterial resistance to phages. So there's a whole slew of things out there that don't necessarily work very well that potentially are allowing bacteria to be at least somewhat resistant to phages. And this is what this talk is going to be all about. And so I'm listing 4 layers of these resistance mechanisms. One has been described as a numerical refuge, another as what I'm calling a probabilistic resistance. There's also what I'm calling phage delay, and lastly there is bacterial escape, which comes from the biofilm literature and is not necessarily something that we want to think about as a phage resistance mechanism, at least up until this point. So numerical refuges, these are essentially can be described as spare the loser. The bacteria that aren't present in high numbers aren't going to support high phage concentrations, and therefore the phages are not going to be around in high enough numbers to wipe out the bacteria. This is essentially the opposite of what has been described as kill the winner. Probabilistic resistance, this is a resistance mechanism that's encoded by a bacterium. It's not necessarily effective every time that a phage infects that bacterium. So there can be leak through. It might be substantial leak through. It might be relatively minor amounts of leak through, but nonetheless leak through. The phages are going to be able to produce more phages in at least some of these infections despite the presence of these resistance mechanisms. In terms of phage delay, we're talking about inhibitions to phage population growth. So these are things that bacteria are imposing upon phages for a variety of reasons that have the effect of reducing phage burst sizes, extending phage latent periods, or causing phages to take longer until they adsorb to bacteria. And then finally, there's bacteria escape. So this is bacteria that are leaving biofilms, and in the course of leaving these biofilms, they are potentially leaving phages. So the bacteria are escaping from phages. Metaphorically, the bacteria are running away from phages. And so these are layered mechanisms of imperfect resistance. And I'll be emphasizing the bottom 2 of these resistance mechanisms. We can describe this as phage delay towards enhancing bacterial escape from biofilms, though I'll be talking about the other 2 at least a little bit. This is something that I published in 2015. This was at that moment, and really still, my thinking about the interactions between phages and biofilm bacteria within larger environments. What we have here is a microcolony. What we have here is a microcolony within the same biofilm that's some distance away. What we have here is a microcolony within the same biofilm that's an even larger distance away. These are all microcolonies consisting of the same types of bacteria. So they have the same phage sensitivity, or approximately so. And this is a microcolony even further away, found in a different biofilm, on a different surface. The phage would have to, and also the bacteria would have to have left the original biofilm in order to reach this new surface of this new biofilm. Now we can map these ideas onto this figure. Remember, at the bottom we have the target bacterium, at the top we have larger environment. We have exponentially increasing volumes, we have decreasing bacterial densities, and also decreasing phage densities as a consequence. And so here we have an individual cell that corresponds to that target bacterium. That's our microcolony there. The biofilm as a whole is here. And this is our larger environment still. So from the phage perspective, in terms of these ecological interactions with bacteria, the phages want to generate enough virions to survive the move from one biofilm to another, to find a new microcolony, a new biofilm, without being inactivated. The more phage progeny a phage can produce in association with the biofilm, the more likely it is that at least one of those phage progeny will survive to find a new biofilm. And from the bacterium's perspective, they want to generate and then release enough bacteria to survive the extra biofilm search to find new locations to found new microcolonies. And as I've noted, this should be a race, especially when a biofilm is being attacked by a phage or has the potential to be attacked by a phage, between bacterial escape and biofilm exploitation by phages. So the idea of a numerical refuge is really what I was talking about with this somewhat blue figure of mine with all the circles. This larger volume creates a situation in which bacterial densities are overall in the environment relatively low. And that is not overall bacterial densities, but bacterial densities consisting of a specific type of bacterium that a phage is able to propagate on. So larger volumes, when you have bacteria that are living as biofilms, because the bacteria are not inhabiting the entire volume, but just the biofilms, larger volumes can reduce bacterial densities, thereby creating numerical refuges. And I would suggest that this is an important reason why, in fact, biofilms are resistant to phages in the wild. That is, they're not necessarily resistant entirely to phages in nature, but instead there just aren't enough phages around to wipe them out because there just aren't enough bacteria of specific types to support enough phages to wipe out these biofilm bacteria. And this is the very idea of a numerical refuge as applied to biofilms. And it's also something that an idea that I published in a paper in 2012. So this takes us to this figure that was published in early 2016. So this represents a microcolony. The little dots are individual cells, and the little green star that's in there and the arrows represent phages. So these microcolonies are not homogeneous. They vary. Even if you have a single species, which these microcolonies, I'm by and large assuming consist of just a single species of bacterium, equivalently in a genetic sense phage susceptible. These microcolonies are heterogeneous. There are parts of the microcolonies that will have denser biofilm matrix, that's the EPS or extracellular polymeric substance, and parts that have less dense matrix. And so the less dense matrix presumably is associated with the newer growth of the biofilm. Similarly, you're going to have regions of the biofilm that have greater levels of metabolism, metabolic activity than other parts of the microcolonies. And in these regions that have the higher metabolic rates are more or less associated with the regions that potentially have the less dense EPS, biofilm matrix, and these are the surfaces of the microcolonies. And these surfaces of the microcolonies just happen to be where the phages are most likely to encounter the microcolonies first. And also because of the higher metabolic rate of these cells, this is presumably where the phages are generating most of their new phage progeny, at least those phages that are exploiting biofilm bacteria. So returning to this figure momentarily, so perhaps most of phage productivity during exploitation of microcolonies is of these peripheral bacteria, that is the surface bacteria, the ones that are found on the surface of these individual microcolonies. But what about the exploitation of the rest of the microcolony? And so my answer to that is that let's talk about phage delay and bacterial escape. So starting with bacterial escape. Oops. So this is a traditional way that people have represented the life cycle of a microcolony. They call it the life cycle of a biofilm, but it's really of a microcolony. We have attachment of a disseminating or dispersal cell to a surface. It replicates and produces more of itself that forms these little tiny colonies. The colonies get bigger. At some point, these microcolonies release disseminating cells, dispersal cells that go on to find new surfaces to initiate new microcolonies. And we can call this bacterial escape. But there's more to bacterial escape than that, so we have a more sophisticated view on bacterial escape. So here we have this bacterial escape that is these flagellated disseminating cells. They're coming out of the center of the microcolony, something called seeding dispersal or central hollowing. We also have bacterial escape that's more passive, where we have an erosion of bacteria from the surface of the microcolonies. And then we can have erosion that consists of not just a single bacterium, but multiple bacteria, clumps of bacteria. We can also have sloughing off of the biofilm as a whole, or the microcolony as a whole, but that's not really something that I'll be talking much about. I also have had an ongoing interest in resistant mechanisms to bacterial encoded resistance mechanisms to phages. And this is what we're showing right here. On the bottom is abortive infections where you have the bacterium dies while essentially, hopefully, killing off the phage, although that doesn't always work. In other words, sometimes the phage isn't killed off. And also we have restriction, which I'm defining as the phage is killed off, the bacterium doesn't die. So restriction modification systems, but also CRISPR-Cas systems are typically, ideally, are displaying restriction. We also have a bunch of other things up here. So there's reduced burst size and delayed lysis, which I call together reduced infection vigor. And then we also have bacteria-imposed delays in the ability of phages to reach the bacterial surface. These can be imposed, for example, by extracellular polymeric substances. So you can think of the circled as mechanisms that can delay the phage exploitation of bacterial microcolonies, either by slowing phage propagation by having reduced burst sizes, extending the infection periods, or increase the amount of time it takes for the phages to reach the next bacterium. And these mechanisms of delay might have the potential to increase the ability of bacteria to escape from microcolonies. So phage delay. So this is a representation of a microcolony. On the outside, you've got the surface bacteria that display a higher metabolic rate. They're presumably more susceptible to phages. And the interior, you have these less susceptible phages. They have a lower metabolic rate. It's harder for the phages to reach these bacteria. So anything that can slow down phage propagation or movement into these microcolonies can interfere with the phage propagation through the microcolony. In addition, anything that could speed up the rate at which the bacteria leave these microcolonies can result in the microcolonies, in fact, being able to reproduce themselves to a greater extent, because if these bacteria can escape, they presumably can escape from phage exploitation. At least that's going on within that microcolony. And the size of these microcolonies might be important, because the region in which you have phage exploitation going on, if the phages can't expand that region rapidly, if it's a reasonable distance away from where the bacteria are escaping, and they might be escaping from all sorts of different places, then that might delay the rate at which the phages can totally exploit the microcolony and essentially infect all of the potentially escaping bacteria. And the whole idea behind this is that the phages are not miraculous necessarily in their ability, once they've reached a microcolony, to immediately wipe out that microcolony. So these can all be viewed as mechanisms of bacterial resistance to phages. So delay of the phage propagation in association with the microcolonies, delay of the phage movement within the microcolonies, but also the ability of the bacterium to escape. And not only is there phage penetration into microcolonies, but there's also the potential for phages to propagate on the surface of microcolonies. So this is another microcolony. This is a phage that's attacking that microcolony. The phage is much bigger than the phage would be in real life, as represented here. So the phage initially absorbs, presumably on the outside of the microcolony. These are the cells that are most available. They also presumably have extracellular matrix that is most easily penetrated by phages. Now, when these phages start replicating, they are not necessarily all the progeny they're released are not necessarily all going to remain in association with the microcolony. They have the potential to diffuse out of the microcolony. And in fact, if there's fluid flow over that microcolony, they have the potential to enter into that fluid flow and be swept away from the microcolony. So the basic idea is that not all of the burst of the initially infecting phage is going to necessarily stick around within the microcolony to further penetrate into the microcolony or to move and exploit this other cells that are found on the surface of the microcolony. It's likely that these phages, in fact, are finding these other cells mostly by adsorbing to the cells that are in the immediate vicinity of a phage burst, of a lysing bacterium, rather than diffusing long distances, that is multiple cell lengths over the surface of these microcolonies. So anything that interferes also with the effective burst size, the amount of progeny phages that are being produced, including the bacterial absorption to bacteria that are to some degree resistant to these phages because of metabolic insufficiencies. So these bacteria might take longer to lyse. They might produce fewer phages altogether when they produce the phages. They also may not produce phages at all because of the bacteria in the more interior part of the biofilm being less able to support phage infections because they have a lower metabolic rate. And the result of this is that there'll be fewer phages available to spread around the microcolony. In other words, to speed up the rate at which the bacteriophage is able to exploit the surface of the microcolony to move about. So basically anything that limits the formation of a plume around the microcolony should slow phage access to the microcolony surface bacteria. So back to bacterial escape. So this again is a microcolony. And what we have are the surface bacteria that are potentially somewhat susceptible. And we have these more interior bacteria, because they display metabolic insufficiencies, they're less susceptible. And then we have the foundation or base of these microcolonies, which may be really far from nutrients and oxygen, and maybe the least susceptible at all. But when you have central hollowing going on, then actually the bacteria that are found in the very interior of the microcolonies can have a higher metabolic rate. And in fact, these are biofilm microcolonies that are displaying central hollowing. What you have are microcolonies that are actually relatively big. They're about 120 or 150 microns in diameter. So these are the microcolonies. That's the central hollowing going on in the center, that grayish part that's within the black border. That actually consists of motile bacteria, or at least bacteria that are in a process of being motile or forming flagella. And you have surrounding them this thing that's called a wall phenotype. And this wall phenotype is actually quite wide. It's 20 to 30 microns wide. And that's, I'm just roughing this, maybe 5 to 10 cells thick. So that is potentially a substantial barrier that the phages would have to cross in order to reach the presumably highly metabolically active motile or forming motile cells in the center of the central hollowing microcolonies. The question is, can they cross that barrier? So we can throw all of this together. So first of all, numerical refugees, this blue circled figure is what I was talking about there. But we can talk about all the other ideas in this one figure here. We have the adsorption on the right of a phage to a microcolony, and then upon infection and release of phage progeny, to some degree we have diffusion of those progeny away from that microcolony, reducing the effective burst size. We also can have resistance mechanisms that don't always work. So some fraction of the time phages are produced, it might be a relatively large fraction of the time. In fact, there are probably biases against publishing on mechanisms that aren't too effective against a given type of phage. So there's likely to be lots of mechanisms out there that have that are not very good at slowing down phages, but still may slow down phage propagation to some degree. We can have reduced infection vigor imposed either by resistance mechanisms that aren't working all that effectively, or by metabolic insufficiency by the bacteria found within the microcolonies. We can also, instead of having these phages penetrating more deeply into the microcolonies, the phages may instead, in fact, be propagating, absorbing and propagating more on the surface. So these are mechanisms that have the effect of reducing the total number of virions that are produced per infection. They're reducing the effective burst size, and therefore reducing the rate at which these phages can penetrate into the microcolony. Now, very important to these ideas is that these microcolonies are densely packed with bacteria. So as a consequence, the phages are going to have a high potential to adsorb to the bacteria. And what they'll do is they'll be released, and they presumably will very quickly adsorb to very close by bacteria within these microcolonies. And these bacteria, especially as you go further into the microcolony, may display a lower metabolic rate. And so as a consequence, the latent periods may be extended, the burst sizes may be dropped. They also, in fact, may not even release phages at all because they are in a more stationary phase state. In addition, the matrix, the biofilm matrix, may be less penetratable by the phages at this point. This will lower the rate of adsorption, but won't increase the rate at which the phages are able to diffuse past these bacteria. In fact, it would slow the rate at which they can diffuse past the bacteria. So the net effect is that the phages are impeded, slowed down in their ability to, in fact, diffuse further into or infect their way further into these microcolonies. And these effects have a compounding effect, so one builds on the other, on the other, on the other. So you have lots of little effects that in combination have the effect of potentially substantially reducing the rate at which phages can penetrate into microcolonies, and indeed maybe reducing it to the point where the phage infections are stopped cold, at least in the interior of the microcolonies. And also, the phages likely are better able to exploit the bacteria, reach and exploit the bacteria that are on the surface of these microcolonies, produce more phages in the process, so that those phages can go on and found new microcolonies. So it's these surface bacteria in particular that the phages may be exploiting to make new phages. The fact that they're penetrating further into the microcolony may, in many cases, just be incidental. It's just what phages can do. It's not necessarily contributing greatly to phage population growth across larger environments. And so this is a transition So what about biofilms and phage therapy? So phage therapy, it's approximately a 100-year-old technique using bacterial viruses to combat especially pathogenic bacteria. And when do we use it? We use it when bacteria are antibiotic resistant to the antibiotics we would want to use against them, or the bacteria can be antibiotic tolerant, that is still sensitive in the laboratory, but in situ, in fact, they're not killed off by the antibiotics. Or we can have reasons that we in fact don't want to use antibiotics because, for example, the antibiotics can have a negative effect on our microbiome. These are phage therapy reports that and the use of phages against infections in humans. So this is clinical phage therapy. A large fraction of these papers deal with the phage treatment of chronic, antibiotic-tolerant, presumably biofilm-associated bacterial infections. Interestingly, if you go through the older literature, and in terms of older, I'm talking about the 20s, 30s, 40s, that's 1920s, 1930s, 1940s, what we are probably seeing is less treatment of these chronic bacterial infections because people didn't have access to antibiotics in the same way they have today. So if they were using phages, they used phages right away. And so one can imagine that treatment of infections, in fact, was easier in those days than it is today because the infections themselves were less mature in terms of, for example, biofilm formation and therefore more susceptible to phages. This is a list published in the middle of 2016 that shows the animal model studies where we had the biofilms, or rather the chronic infections, or rather the infections by the bacteria, form over at least a 24-hour period before the phages were applied. So presumably these are biofilm associated. And by and large, using phages against these infections has been quite effective. But I would argue that really we are seriously lagging in our preclinical exploration of phage therapy, of especially challenging to treat chronic bacterial infections. So given at least the potential for phage delay as a mechanism of bacterial, or as mechanisms of bacterial resistance to phages, how might we guess what would be relevant in the clinic to treat these, especially mature chronic bacterial infections that are probably biofilm associated? So these are a list of impediments that these biofilms are imposing on the phages. A mature biofilm matrix, stationary phase bacteria, reduced infection vigor imposed by various means, including low metabolic sufficiency by the bacteria themselves, perhaps virion losses at the treatment site, not all of your phages are getting where they need to be, delayed penetration of the biofilm by the phages, and also anti-phage patient immunity, although that lately has been shown to be perhaps not as big a deal as people might like to think it is, the latter one. And how do we deal with these things? We deal with debridement, which is basically attempting to get rid of much of the biofilm and necrotic tissue before we in fact add the phages. So the phages have the potential to reach the biofilm bacteria more effectively. We use sufficient phage numbers. We're not necessarily going to count on the phages to be able to in situ increase their numbers sufficiently. We're not going to add 10 to the 5 phages per ml and assume that the phages are simply going to replicate to sufficient numbers. And for sufficient numbers, by the way, is about 10 to the 8 per ml, whether you're achieving that through adding phages solely or you're achieving that instead through the phages replicating in situ. Multiple or continuous dosing. We're not going to count on just a single dose because, in fact, the phages may not be able to sustain themselves against these biofilm bacteria with a single dose. You may have to keep adding phages, keep resupplying them to attack especially bacteria that have become newly available to these phages because of this next one, the fourth one, because phages are able to, in fact, lyse bacteria. In fact, in treating biofilms, it's unlikely that using phages that don't lyse bacteria will be as effective as using ones that do. We can also potentially use antibacterials such as antibiotics as long as they're not interfering with phage replication. And then there's EPSD polymerases, which may make bacteria within biofilms more available to phages. Although EPSD polymerases tend to be highly specific in terms of the EPSs, the extracellular polymeric substances that they are able to attack. And so there are limitations in terms of our ability to rely upon EPSD polymerases for attacking biofilms. And so that's the end of the talk. I want to make sure people know that I have a website page, mytalks.phage.org is how you reach it, that basically has a lot of my recent talks because I've been recording them and posting them online. And otherwise, that's the end. Thank you to the organizers. Thank you to David Awe, who helped put together all of this equipment that I'm using to record this talk. Thank you to Laura Applegate, who pressed on the screen of the video camera to make sure that I'm in focus throughout this talk. Thank you very much.